Good afternoon, everyone. My name is Kyle Jarrett, and today we're going to be taking a look at the lung microbiome, friend or foe, specifically the lung microbiota in correlation with malarial disease. This was a research paper conducted by Mukherjee and their associates in a paper that they titled Host Lung Microbiota Promotes Malaria-Associated Acute Respiratory Distress Syndrome. This disease, which is known as MARDS, occurs in severe forms of malarial infections. This is an important area of research as malaria is one of the most dangerous diseases impacting our world today. But we also know very little about a part of our own body, that being the lung microbiome, and how it is affected when the host is infected. The authors of this paper aim to answer this question by infecting mice with two strains of the Plasmodium parasite, which is the cause of malaria. Upon which, after about a week or so of the infection, they would examine the lungs of the mice to see how they had fared. They would come to discover that the lungs of the mice were similar to those of humans whose cause of death was indeed caused by MARDS. Before we really dive into this paper, I'd first like to introduce you to some key terms. I'll give you a minute to overview them. So before we dive into the paper itself, what's the deal with the lungs specifically? Well, the lung microbiome is a highly under-researched area of the human microbiome today, and is arguably one of the most important areas that should be studied. Without healthy lungs, we cannot receive the amount of oxygen that we need to survive, and the human body becomes weaker, resulting in death for many cases of those with lung diseases. So how does malaria come into play with the lung microbiome, you may ask? Well, there's only one way to find out. So come along with me and learn what I learned. So before we get into this paper, I'd like to just give you a brief overview of the gut versus the lung microbiota so you get a little bit of an understanding of what we're looking at. Well, the gut microbiome, what we'll start with, is made up of a variety of bacteria, as we'll come to learn in this course, such as fungi, viruses, bacteria as stated before, and various other organisms that find their way into this area and survive. Normally, the gut microbiota is dominated by a specific group of bacteria, and it usually stays that way unless it becomes altered. This is known as dysbiosis. The lung microbiota is not so different, though, as was previously believed. It was initially thought that the lungs were a sterile area of the body, but now, after 16S ribosomal RNA sequencing, it has been discovered that the lung microbiota is actually similar to that of the gut microbiota. The most common phylum found in the lung microbiota includes those of bacteroidetes and firmicutes, which are the same as the gut microbiota. It is also known that the gut microbiome interacts with the host immune system, as does the lung microbiome. The gut and lung microbiome are also connected to each other, however, through what is known as the gut-lung axis. This allows both the gut and lung microbiome to interact not only with the immune system, but to help determine its responses so as to avoid damaging the host itself while removing any unwanted pathogenic organisms. As we can see here, we can see that both the gut and lung axis aid in interaction with blood and the vagus nerve and oropharynx. Significant alterations in the gut microbiota occur during the respiratory diseases, so we can see how they're connected through this axis as well. And we also see that while the gut has high microbial density and high microbial diversity, we learn that the lung has very low microbial diversity. Looking ahead, we're going to look at malaria as one of these pathogens that is involved in this. So, malaria can take on many forms, with clinical manifestations developing, some which can be more severe than others, as we will later discuss in this presentation. And this all depends on the strain of the parasite that one becomes infected with. Malaria is a disease that is transmitted by mosquitoes possessing the Plasmodium parasite. The parasite enters a human host through the bite of an infected mosquito, where it will pass through the bloodstream. The parasite will make its way into the lungs of the host, where it spreads and begins to infect red blood cells, thereby spreading its way through the host circulatory system. While this disease is extremely dangerous on its own, and can be worse depending on the strain that someone becomes infected with, it becomes even more of an issue when the microbiota of the lungs begins to aid in the severity of the disease rather than aiding the immune system remove it. This is where our journey of the lung microbiome will take place. with it aiding the malarial disease and becoming more than just a blood parasite, but a respiratory nightmare. So, severe malaria can result in a multitude of disease manifestations, normally resulting in death. These can include respiratory distress, which this presentation will cover more on specifically, multi-organ failure, and cerebral malaria, which is better known as a coma. It is currently unknown exactly what causes someone to be afflicted with a more serious ailment compared to those with milder cases. This is currently being researched, and we will hopefully have more answers sooner rather than later. To perform the experiments conducted in this paper, they used rodent models to showcase how the host microbiota is a contributing factor towards the onset of respiratory distress, as well as increasing the chances of the host dying due to the infection known as MARDS. Due to the parasites isolating themselves in the lungs of the host immune system becomes sustainably active, thereby not only weakening itself, but harming itself in the process due to the constant inflammation being produced. This is caused by the production of interleukin-10, which is a common anti-inflammatory cytokine that is produced by T cells as part of the innate immune system. This is used in turn to reduce the inflammatory response as well as microbial control. This has an unintended side effect, however, as this also affects the natural microbiota of the host, thereby resulting in severe lung disease. So with the inflammation, if you're not only trying to get rid of the bad bacteria that are in there, but you're getting rid of your good bacteria, you're not really doing yourself a favor, because then, while the good bacteria dies out, the bad bacteria that survive the inflammation can then take that place, and in turn are able to cause the disease. In a quote from the article, they state that the host's anti-inflammatory response to limit tissue damage can result in loss of microbial control, which promotes MARDS. There may be a little bit of a safety catch though. By using linezolid, which is a currently used antibiotic that is available for controlling lung-associated infections, the researchers noted that it was able to prevent deaths caused by MARDS. So there could be hope. So let's take a look at these disease manifestation correlations, which may tell us why some people develop a more severe clinical outcome than others. One of these manifestation correlations would be host genetic factors, which influence red blood cell characteristics, as well as the VAR gene seen in the Plasmodium parasite itself. It is unknown still, though, why someone infected will develop a greater chance of developing a certain condition, such as severe anemia rather than cerebral malaria, but this is research being done today. These diseases, however, also show a correlation with the intensity of transmission in the infected person's surrounding area. The infected person's age is one other correlation factor. ARDS, which would be acute respiratory distress, is one of the most severe malarial disease forms that a person can develop. Around 20 to 30% of patients with a severe case of malaria will develop MARDS specifically. Interestingly, this begins after the malarial treatment has already been administered, rather than prior to administration. It is well known today that dysbiosis of the gut microbiome is associated with an array of disorders. The host microbiota contributing to malarial diseases, however, has only just begun its research. Two independent studies both found that by using two different rodent models, as well as two different malarial strains, both resulted in the same outcome, that being the gut microbiota becoming altered from its original form. Something that has been discovered in alterations of the bronchopulmonary tract microbiota of the lungs specifically is that it has the potential to exacerbate diseases such as cystic fibrosis and COPD. This is just yet another example of the natural microflora of the human body acting in tandem with a pathogenic organism to result in further damage to the host. If we take a look at our figure here, looking at the left figure of the lungs in the red with dysbiosis, we can see that pathogen multiplication, as well as altered microbiota resulting in reduced diversity, as well as tissue damage can also result in dysbiosis of not only the gut, but the lungs, aiding in infection results. However, there is microbiota restoration, which is able to in turn prevent that from happening as well as correct it, which can boost immune response involving pro-inflammatory and anti-inflammatory reactions by the host immune system to not only clear the pathogens, but as well as resolve tissue damage and result in microbiota restoration, which would then resume normal diversity of the microbiota, allowing the host to recoup and reheal. Now, if you recall previously, I did say that there was a safety catch associated with this. And that's true. There is a treatment for MARS, kind of. While there is the antibiotic that is known as linezolid, which is currently available for controlling lung-associated infections, it becomes ineffective in preventing MARS once dysbiosis of the lung microbiota occurs. Now, if you give this to someone infected with malaria before dysbiosis of the lung occurs, they should not develop MARS. However, once these symptoms develop, it is already too late. This is similar to the rabies vaccine, which also becomes ineffective once symptoms develop. So if someone gets bitten by rabies, you normally have, I want to say, about 2 weeks or so, if I remember correctly, to get the vaccine, and then it will clear out the infection. However, once these symptoms develop for rabies, there is no cure. So how exactly was this research accomplished? Well, they used an aluminum MiSeq benchtop sequencer, which we can see in this figure over here on the left, to analyze the 16S ribosomal RNA that was taken from the mice samples. Using Illumina for this area of research is a good start, but thankfully was not where they ended, as the journal highlights the use of other methods. These other methods included Kraken-2, which was further used to analyze the important data at hand. This is critically important, as while Illumina is cheap and does provide high-quality bases, it provides extremely short read lengths, making genome assembly difficult. This is highlighted in our Module 1 Lecture 3 slides, where it states that assembling a genome from short sequences will increase the error rate. After using Illumina to sequence the bases, the authors inputted the FASTQ files into a data2 pipeline. Once the files were further trimmed, they were used to create an amplicon sequence variant table, or ASV table, where the chimeras were removed so as to only focus on the purest strains available. And if you don't know, chimeras are like a jumble of stuff together, so it would be like different strains together, not a pure strain, which would result in more, not linear DNA or samples. You'd rather have the pure strains so that you know the best samples to look at so you can get the best outcome for research. Samples from the lungs were also taken themselves after the parasites were removed first. These samples were taken so that they could measure the cytokine and chemokine levels, which are important in informing us of the degree of the immune response. Bronchoalveolar lavage samples were taken for sampling as well. So let's take a look at the results of the experiment, shall we? By infecting 2 separate rodent models with 2 unique strains of the Plasmodium parasite, which as we know is the cause of malaria, they were able to showcase the damage to both rodent-type lungs. Both samples showed a high similarity to those of a pair of human lungs that had been damaged by MARS. To test their hypothesis of whether the lung microbiota was associated with the development of MARS, they plated a sample of each mouse on the 3rd and 5th days post-infection. This was done to allow MARS to begin establishing itself within the lungs. They would plate once more on day 7, as this is when MARS begins to actually develop symptoms and show itself. On day 3, they noted no differences between the mice strains. This would change, however, on day 5, where the PB-K173 mice, PB being Plasmodium berghei, which we can see in this image on the left, these mice showed a 2 to 3-fold increase in their CFUs, which is their colony-forming units. This would again increase by an exponential amount on day 7 and would increase by a 9 to 10-fold. Once this data was collected, 16S ribosomal RNA gene sequencing was conducted on all 3 samples of mice, those being the PB-K173 infected mice, the ANCA infected mice, as well as the non-infected mice, which served as the control group. It was discovered that on days 5 and 7, the PB-K173 infected mice, which showed an exponential increase in bacterial load as we just discussed, had their normal lung microbiota composition altered. Something else of note was the increase of E. coli as well as staph bacteria. In the non-infected mice and the ANCA-infected mice, an increase of E. coli and staph species in the lungs has been previously associated with pulmonary disorders. In turn, the other bacterial families that comprise the lung microbiota showed a relative decrease in conjunction with the increase in E. coli and staph. This shows us that as the bacteria that are normally not supposed to be there, like E. coli and staph, as they increase, they take up the room that the normal bacteria that's normally there would naturally be in, which then results in worsening of the infections. There is a high level of correlation between increased parasitemia levels among day 5, as well as with the mice who die from the complications associated with MARS. This is something that we'll see in a figure later. This result led to the development of MARS. After performing a bacterial family abundance analysis, it was noted that the infected mice with the PBK173 and ANCA strains both saw major changes to their lung microbiome compared to the mice who were prevented from becoming infected. We will have a chance to look at a bacterial family abundance analysis later in the slides. One final test that the researchers conducted involved infecting germ-free mice on specific pathogen-free mice. So when they infected the GF mice and the SPF mice, they noted that all of the SPF mice that were infected with the PBK173 strain died of complications from MARS on day 7, as was expected. Of note was the amount of GF or germ-free mice that were infected with the PBK173 strain as well, but who never developed MARS. Those that did, however, develop MARS died due to the complication associated with it, but at a later time. These germ-free mice that died from MARS actually died around day 10 rather than day 7, which goes to show that the microbiota of the lungs does have an impact on the disease lethality, because these specific pathogen-free mice do have an actual lung microbiota as they do a gut microbiota, and they died on day 7 just as the other mice did. Whereas the germ-free mice have no microbiota whatsoever, and while they did die from MARS, from those that did, they did so 3 days later. This is figure 1 taken from the paper, and in this image we can see the correlation between bacterial load and oxygen saturation, which is how much oxygen is within the red blood cells making their way throughout the body. In section A of this figure, we see how the C57BL mice, when injected with the PBK173 strain, have the lowest oxygen saturation levels. This is to be expected, as this strain was the deadliest of the two used in the study, with those infected with this strain dying around 7 days post-infection, as we can see here with their oxygen saturation levels plummeting in between day 6 and 7. It can also be seen in section C of this figure that as post-infection days increase, so do the CFUs. This allows the reader to visualize the correlation between the CFUs growing in size, resulting in the oxygen saturation depleting. So as we see in the CFU column in section C, as the colony-forming units increase, the oxygen saturation decreases, showing that as the bacterial load continues to mount, the less oxygen the red blood cells can then activate themselves. And if we compare the C57BL mice compared to the DBA2 mice, we see that those non-infected showed no difference between them because they were never infected, whereas the ANCA mice did have a drop in oxygen saturation, but only to about a 86 to 88 degree percent compared to those infected by PBK173. This is section D of figure 1, and this is a picture of our bacterial family's presence and absence within mice strains, which I'm sure you have all been dying to see. So each row is a bacterial family, and each column is a mouse. Something to note from this figure is the bacterial families that are absent in PBK173 mice compared to those infected with the ANCA strain and those who are in the non-infected group. So if we look here, we see that in the K173 area, they are missing a lot of species such as Ruminococcacea, Oscillosperacea, and even some Micrococcacea, whereas the non-infected ANCA strains are very high in Streptococcacea and Enterobacteria. Now let's take a look at figure 3 of this article, which is titled SPF versus GF parasitemia load with survivability percent. If we look at section A on the far left, we can see that the survivability rate of the SPF mice with the PBK173 strain reaches 0 around the 7 to 8 day mark, as was mentioned before. It is interesting to see that while the parasitemia rate for the germ-free mice does continue to grow, they do not exhibit nearly as high a lethality rate compared to the SPF mice. Taking a look at section C of the figure, we see the survivability rate for both types of mice drastically plummet around the 6 to 7 day mark when they are infected with the Onca strain. Something you may have also noticed within these figures is these red asterisks on these long tissue samples. These are associated with the development of severe alveolar edemas and are only present in the BBK173 infected SPF mice, not in the germ-free mice. These examples are shown here to provide a visual aid of how the microbiome of the lungs is associated with worsening malarial infections, resulting in the development of MARDS. This is proven by the SPF mice not only showing the lowest survivability rate, but also being the only strain of mice to showcase the development of severe alveolar edemas. When the mice were treated with linezolid, MARDS was prevented, however. ECM, being experimental cerebral malaria, was not prevented. Linezolid, however, was unable to prevent MARDS once dysbiosis of the microbiome had occurred. However, treatment with linezolid, beginning about 3 days post-infection, which would be before MARDS was ever developed, was able to prevent mice from developing it. The article states that they have data that without a shadow of a doubt proves what the cause of MARDS is. They state IRBCs, those being infected red blood cells, sequestration in the lung, along with concomitant local inflammatory changes, have been previously proposed as a main cause of MARDS in humans. This is showcased by the quote above in this figure, which is figure 2 from the article. It can be seen in part C of this figure that the group with the highest CFU is that of the Onca strain with the highest parasitemia rate. Figure B serves to show the parasite sequestration in the lungs of the DBA2 strain of mice. This section of the figure shows that 5 days post-infection, the DBA2 strain of mice with the highest parasitemia rate has the highest sequestration compared to those with a low parasitemia rate or those of the non-infected strain of mice. One way to prevent MARDS, as discussed in this paper, is the use of antibiotics that would control alterations of the composition of the microbiota. Something that I wonder is where the malaria vaccine would also be able to prevent MARDS from developing as well. The lung microbiota is not the only microbiota to assist in worsening infections, however, as I'm sure we'll learn more in this class or perhaps through somebody else's presentation, that the gut microbiota is also associated with infections. Gut-associated bacterial families are shown to be overpowering the normal microbiota of the lungs in ARDS patients who typically die due to the disease. Interleukin-10, or IL-10, aids in worsening the infection as well by reducing the bacterial load of the lungs. While this is supposed to prevent pathogenic microbes from establishing a foothold in this area, it in turn kills the good bacteria already residing within the lungs. This attempt at protection of the lungs by IL-10 instead results in the lungs becoming overpopulated by the pathogenic microbes, resulting in the lungs becoming overloaded by microbes and MARDS developing. So why exactly did I choose this paper of all of them? I chose this article because I wanted to learn more about how the human microbiome interacts with the immune system of the host regarding an ongoing infection. I also chose this article specifically because I was unaware of the existence of the lung microbiome. I knew of the gut microbiome, its interactions with the host immune system, and how dysbiosis of the gut microbiome can affect the body's natural immune response. I was unaware, however, of the lungs microbiota, as I initially believed the lungs to be a sterile part of the body. While this is not the case, very little is still known about the lung microbiota even today, and why it is a high topic of research today, and hopefully more will learn about it in the future. In conclusion, this paper showcased how the lung microbiota is similar to that of the gut microbiota in aiding the worsening of infections unintentionally. Due to the host immune system trying to fight off the infection, it in turn aids it instead. By showing how the disease interacts with the host microbiota, it allows us to determine new treatment options that prevent this from happening in the future. It also allows us to develop better responses to these outcomes so as to prevent them from occurring in the future. I hope that this paper, as well as this presentation, was as interesting to you as it was to me. With time, we may see new treatment options develop not only for treating MARDS, but malaria as well, as it is one of the most dangerous parasites in the world today. Maybe one of you will be inspired to join the research efforts regarding malaria or the lung microbiome, and be the one to uncover the breakthrough that changes everything. Maybe this year in 2025, it will be the year that the lung microbiome has its explosion in terms of discoveries, just as the human gut microbiome was years ago, or the human genome sequence from 2001 to 2006. Only time will tell, however. Thank you so much for watching. Before you go, though, I do have a few questions that I have asked. Would the malaria vaccine be capable of preventing MARS from developing? If we know that it has a 40% success rate in preventing malaria, because the malaria vaccine is not fully effective, would it be able to prevent MARS from developing, though, when it does in turn prevent malaria? Or would it not be able to prevent malaria, but then be able to prevent MARS itself, though? Question 2 is, are there any other antibiotics that may be effective in preventing MARS if the disease becomes resistant to linezolid? As we know, antibiotic resistance is a massive topic in not only medical talks today, but in any type of infectious disease classes, virology, bacteriology, or even maybe in this microbiome course. With antibiotics becoming less and less effective in treating diseases, are there any other antibiotics that are currently not used in lung infections that may end up actually be able to work, like those that are associated with like gut diseases? Or are there new antibiotics that we could put together or just completely create ourselves that are brand new that could also be used in treating this? Question 3 is, how can we treat the disease without affecting the microbiome of the host? As we've discussed extensively in this paper, altering the gut microbiome or the lung microbiome of the host results in not only the good bacteria being affected and potentially dying off, but that in turn allows the bad bacteria to then take over that place, resulting in the infections worsening. Is there any way that we could have selective effects that only affect the bad bacteria while not harming the good bacteria? This is something that would take a lot of time and effort and research to test. Question 4 would be, what aspect of the lung microbiome may be the most pertinent to study today? I personally think that it is what bacteria and organisms already comprise the lungs, as we don't know much about what really comprises it. We know that there's bacteroides and firmicutes, but are there any other organisms there that we haven't yet discovered that could belong to a different phylum, maybe a different clade, or just different species of these that exist that we have yet to discover as well? And lastly, with question 5, if the lungs and the gut microbiota are made of the same microorganisms, such as bacteroides or firmicutes, does that mean dysbiosis of the gut microbiome can result in dysbiosis of the lung microbiome and vice versa? So if we have an issue like intestinal bowel disease or intestinal bowel syndrome of the gut, does that in turn go through the gut-lung axis and have an effect on the lungs? Or does an infection with the lungs, like a cough or something, when you do that, does that in turn then travel back from the lung to the gut axis and then involve dysbiosis of the gut microbiota as well, resulting in 2 separate infections potentially occurring at once? And these are the references I used. I hope you enjoyed this presentation. I hope you learned a lot from it, and I hope I inspired you to not only learn more about the microbiome itself, but maybe the lung microbiome, as you could be the one to make the ultimate discovery that then leads to not only maybe the treatment of MARS from ever developing, but maybe new antibiotics or new treatments in general that could lead not only to lung infections decreasing, but maybe that in turn would have an effect on the gut microbiota as well. So good luck out there in the future.